Welcome you to the 20th webinar of the online edition of the Protelec Electrostatic Conference. The speaker of today is Emil Alexov from Clemson University. Emil got his PhD in physics from Sofia University in 1990. After that, he switched to biophysics and became a staff scientist at the Bulgarian Academy of Sciences, then a visiting fellow at RIKEN in Japan, then a postdoc in City College of New York, a senior scientist at Columbia University, New York, and then in Clemson University. Among his research interests, there is Delphi, electrostatics, development of methods and tools for predicting effect of mutation on protein DNA binding and stability, and predicting pathogenicity of human DNA variants. So the title of his presentation of today will be on the role of electrostatics in molecular recognition. So welcome, Emil. The podium is yours.
Thank you, Walter. Thank you for the introduction. Okay, we already practiced with Walter, and I assume that everybody can hear me and see my presentation. As Walter said, today I'll be speaking about electrostatics in molecular recognition. And this slide perhaps will explain what I mean in more detail. So on the left-hand side, just a second, maybe I will do the trick. So here's my laser pointer. On the left-hand side, you can see a hypothetical presentation of a ligand shown in 4 copies that supposed to be binding to this receptor. And with these tiny blue arrows, we show possible trajectories that the ligand will follow in order to bind to the active site of the receptor, for example. So by recognition, we mean this process when the ligand and the receptor are not bound yet, but they are sensing each other. And eventually we like to be able to model what is the trajectory the ligand takes in order to get into the receptor. How successful is this? To be able to predict what are the conformation changes associated with this process, not just the final physical docking. And to illustrate that this is important in terms of electrostatic contribution to the recognition process, I'm showing on the right-hand side 2 snapshots of a case that I'll illustrate later on in more details. But for now, consider that this is the receptor. This is still the receptor, the very same receptor. This is piece of a macrotubule. And the ligand is the binding domain of a particular molecule. Crystal structure does exist. We simply took the ligand and offset it from the crystallographic position at distance 10 angstroms or distance 20 angstroms. Then we calculated electrostatic potential distribution, and using VMD, converted this distribution in electrostatic field lines. So the thing that we are trying to say with these 2 figures is that even the ligand is offset by 10 or 20 angstroms, there are very strong interactions between the binding partners, which are indicated here by the density of electrostatic field lines and their polarity. Red is minus, blue is plus as usual. So at 10 or 20, even more pronounced, 10 angstrom distance, electrostatics is only force that is not 0 in magnitude acting between the partners. So what I'm going to do first to introduce one of the tools that we use for that study, namely it's called Delphi force. For people that are in protein electrostatic calculations and final difference methods, this perhaps is a boring slide, but essentially we like to be able to have, let's say, a charged molecule or a quite large entity as this piece of macrotubule. A ligand, the ligand could be a small molecule, could be a protein or a domain of a protein, and to calculate what are the forces acting on each of the atoms of the ligand due to the presence of charge receptor. I mean, the atoms of the receptor are fully charged according to a particular force field. The caveat is that we like to compute these forces on the atoms of the ligand, having in mind that ligand is not a water phase. Ligand is also the electrical cavity with a low electric constant. So more details can be seen in the original paper, and this is the link to the web server that lets you calculate such forces between two user-selected entities. Receptor ligand, receptor small molecules, receptor DNA, etc., etc. So to illustrate the importance of electrostatics in a variety of biological cases, here I present one set of examples indicating the direction and magnitude of forces computed between two proteins that serve as an adhesion partner between cells. Co-modulin case, a protein bound to DNA, protein bound to RNA, and favorite think of Marilyn Gunner, reaction center protein. The thing that you're supposed to see, if you have sharp eye, are the corresponding arrows that will indicate the direction of the forces. So in this case, we charge one of the partner, and that is shown here as a non-color presentation. And we compute what are the forces on each atoms of the other partners. These are very tiny red arrows that you most probably won't be able to see, but with the green solid arrow, we show the total force. The thing that I like to point out to you that in all these cases, the force is attractive, pointing towards the bionic partner being a receptor, for example, except in case of co-modulin. And people working in that area know that co-modulin lobes, they tend to be separated of each other before the binding, and indeed the total force is pointing towards extended conformation. And the last example that I like to outline very briefly is the QNON case in bacterial reaction center. With this arrow, we show what is the pulling force of electrostatics to get QNON B outside of the reaction center protein. So this is these examples illustrate the directionality of the force. What about something else, which I will show in the next slide? Namely, we took several well-known examples where electrostatics play a significant role, namely case of Barney's bar star. Kinesin microtubule might bind to the smallest unit of microtubule called tubulin dimer. And the same thing when the microtubule binding domain is taken from another protein, diamine. The thing that we do is to take all these binding partners. This is the thing that I'm showing with my laser point, and to rotate them on 90 degrees with respect to the crystallographic position. So obviously in that case, the interface orientation is totally wrong. However, if you compute what is the torque generated by the receptor protein, the torque is always in direction such that it tries to rotate back the partner into the right orientations of interfaces. You can see it here, there, and there. Now, introducing the two, I will be discussing today two general cases. One of them will be to understand how electrostatic force evolves during a possible trajectory of the partners approaching each other. For example, take a look at this example when the partners are shown with blue and red. And we're going to offset the red in this direction. And then we're going to follow what is the evolution of the force of these two partners, either approach each other or separate each other. And the very same thing, and that will be just a rigid body protocol. We are not going to apply molecular dynamic simulation. Or the very same thing, as I illustrated in previous slide, this is the tubulin dimer, and we're going to offset the binding domain of diamine away from the crystallographic position. And we're going to show what is the change of the force. But in that case, we also allowed for a relaxation for molecular dynamic simulation to sample different conformation state. The second part of my talk will do something that we recently developed called Delphi Force molecular dynamics steered simulations. And this is an important thing according to our understanding, in case when you don't know the bound position, the binding pocket might be closed. So that will require a relaxation of the structure. And that will also allow for conformation changes to be predicted. And also we can also assess what is the most probable trajectory the ligand takes in order to bind to the receptor. Here's the first case that we are going to consider. And actually, that was the motivation of paying so much attention on electrostatic forces. First, I like to and this thing is not working. So you're supposed to see a movie here, how kinase.
Probably you should change from laser back to the pointer in order to make it start.
I see.
I'm not sure, but may go to.
I don't know how to do that.
The pointer option. Let's see the. I no longer see the. I don't know.
I have quite many options, and
I do not see the automatic. Go to automatic.
Okay,
yeah.
Thank you, Walter.
Sure.
So this is a kinesin walking on a microtubule, carrying a huge cargo. And the thing that we like to model is, of course, not this huge ensemble of proteins, but just how the feet of the kinesin step on the microtubule and what is the role of electrostatic of guiding their steps. So I am not going to switch to laser point option. I'm going to use my cursor. Namely, we're going to model the microtubule as a segment of quite many tubulin dimers shown with different colors. We are not totally crazy to model the entire microtubule, which is 1,000 of angstrom in length at minimum. So we're going to consider just a small segment. And this is the dynein domain, microtubule binding domain. We are not going to consider the entire structure of dynein. and actually, this figure does not show the entire structure. There is something more than that. So how we're going to do that is using something that we call the multi-scale approach. Namely, this is our model of microtubule, and then the binding domain of dynein is going to be placed on the crystallographic position and then offset up, left, right, and rotated in order to explore the conformation states. And if you follow these figures, you'll get some sense how we generate the conformation space for the ligand. And at the end, this resulted in about 36,000 different structures. So for each of them, we do a calculation of electrostatic component of binding, namely solvation and Coulombic interactions. And also we take into account the van der Waals interactions for each position. In terms of electrostatics, since it is long range, we use this multi-scale approach, namely the first round, the electrostatic is calculating the entire box that contains the full segment of microtubule. And after that, we do a focusing. The thing that I don't want to get much in details, but I like to mention it at least, is that the focusing technique was modified in Delphi to allow the thinking where we calculate the potential, namely different position of the ligand, not to be taken from the same parent run. So if I change the position of the ligand from here to there, for example, I don't have to do the first run again. We have an interpolation procedure, which, excuse me, makes the calculation of 36,000 cases quite doable. As a result of this simulation, we've noticed that no matter how we offset the microtubule domain, always the electrostatics points to the right binding position as seen in a crystallographic structure. And that also tells us that electrostatic is not only attracting the binding partner to the right position, but also takes care to correct for the orientation and the position if the orientations and positions are not correct. Further proceeding with this case of diene microtubule binding domain to microtubule, we wanted to explore what is the role of something that typically people don't pay attention to. So if you have a sharp eye, you will see this tiny thingy sticking away from tubulin domains. These are called E-hooks. Why they are called E-hooks? Because they are made mostly of glutamic acids, and they are very flexible, and they are not seen in the crystallographic structure. So then our question was, what is the role of these E-hooks, since they are so flexible, in guiding the stepping of diene microtubule domain on the microtubule? So we used the same procedure as I explained to you on the previous slide, namely, we took the original position of microtubule binding domain shown here with red, and we, in stepwise manner, offset it away from that position in vertical direction. And the goal was to see how this unstructured E-hooks shown here with a kind of grayish color, and I'm following them with my cursor, how they contribute to the binding. Again, to reduce...
Sorry, can you speak a little bit louder, please? A little bit.
Okay. So to avoid the complexity, we did a preliminary study to figure out how big the fraction of microtubule should be in order not to skip any possible contribution. And by contribution, we mean ability of some of these E-hooks to interact directly with the binding microtubule domain. It turns out that in order not to omit any possible contribution, we need to take into consideration only two tubulin dimer, this one and that one, and corresponding E-hooks are labeled as A, B, C, and D. Here, very quickly, I'm going to show one movie, kind of difficult to see because of the dark background, but this is one E-hook, another E-hook, third and the fourth E-hook. And this movie is done of the tubulin dimer without the presence of the binding domain. And perhaps you can appreciate that the E-hooks are sampling random conformations, sometimes adopting alpha helical conformation, sometimes just totally extended. But they don't stay in a fixed conformation. I'm gonna stop this thing and then show the same type of modeling, molecular dynamic simulation, in the presence of the binding domain shown here with the blue color. Perhaps you can appreciate that some of the E-hooks, they kind of approach the domain, bind, and then release, bind and release. So this is a process of constantly exchanging state from bound to unbound state. Again, to save time, I'm not going to show the entire movie, but I like you to appreciate that this is a dynamical process. Too much details, but this is the last slide that I'm going to show with so many diagrams. In this slide, I like to illustrate what are the number of contacts shown on the vertical axis as a function of time in case of one of the E-hooks, which we label as letter B, contacting the corresponding microtubule binding domain, and the same type of graph for the other E-hook called E-hook D. Different colors indicate the number of contacts taken at the crystallographic position of microtubule binding domain, 15 angstrom away, 5 angstrom, 15, 25, 35, etc., up to 55 angstrom away. The take-home message is the following. This is really a dynamical process. No matter how far away the microtubule binding domain is, there are still binding events, even at 45 angstrom away. There are some binding events. And also you can also appreciate that For some distances, the binding occurs relatively early, then disappears, again appears again, etc. I'm going to skip this slide because I'm running out of time. And the thing that I like to discuss in more details is this very busy slide. First, I don't like to underestimate the audience, but doesn't hurt to repeat what is the scenario of induced binding. So here with a cartoonish diagram, we like to illustrate that in the free state, both the ligand and receptor might be sampling different conformations. But let's make the situation very, very simplified. Say that in the free state, the ligand takes this conformation 99% of the time, and only 1% of the time sample this conformation. Similarly for the receptor, but when they bind, it is considered that the occupancy will shift, and now this and that conformation will be 99%, the other conformation will be not sampled at all. We decided to probe if we are capable of seeing modeling and seeing such an event in case of a microtubule binding domain binding to microtubule. And I always have troubles explaining this graph, this table and then this graph, but I will try one more time. Here we have the following measurement. We cluster the three E hooks into 1, 2, 3, 4, 5 different clusters and calculate what is their occupancies. So cluster 1 in free state bears occupancy 33%, cluster 2, 19%, etc. So from here to there, the percentage of occupancies decreases. Then we do the very same thing, but in presence of the binding domain. And we identify different 5 clusters with different occupancies. And then we compare the representative of the most occupied cluster in bound state versus the most occupied cluster in free state via RMSD. Obviously, they are quite different. However, the least occupied cluster in free state is very similar to the most occupied cluster in bound state, and vice versa. So then, if you take into account the number of cases and draw a graph, so vertically is the population or the percentage of the E-hook in a bound state, and horizontally is in the free state, you see very nice inverse correlation, which is exactly what we imply in understanding of induced fit binding. And lastly, regarding to investigation of binding domain binding to microtubule, we decided to plot what is the magnitude of electrostatic force over here as a function of the distance between microtubule binding domain and the microtubule. As we anticipated, we saw, of course, the force is practically zero when the distance is relatively large, and then the force getting larger and larger. This is in piconewtons, getting to about 5 piconewtons at relatively small distances, and then becomes repulsing force at the crystallographic position. We were kind of surprised of such an evolution of the force as a function of the distance, but then we decided to think what is the meaning and how useful from biophysical standpoint such a profile might be. And indeed, with this movie, I like to show how such a profile will result in approach of two entities. At the beginning, they approach each other quite fast, and at the end, when the physical bonding will supposed to occur, they slow down. And we decided to call such kind of contribution of electrostatic to the binding process soft landing, namely the biological units, the receptor and the ligand, reduce their speed as they approach each other, and perhaps biologically that thing makes sense. You don't want to crash, like a car crash, the entities when they are approaching to each other. Instead, you prefer to have a soft landing. And very quickly, that prompted us to do investigation on larger case of protein complexes that were taken from Ray Wu, precompiled data set, University of California at Irvine. And we simply decided to take the crystallographic position shown here with blue and red, and to pull out one of the partners in desired position and to study how the profile of the electrostatic force depends on the distance. I'm really running out of time, so skipping the procedure of how to identify what is the right direction to separate automatically binary partners, and happen to be not very easy thing to do in case of protein complexes with irregular binding interface. So this is technology that I'll be glad to discuss with you if there are some questions, but for now I'm gonna skip it. And after performing studies on the Ray Wu dataset, we identified 4 main classes of profiles, namely the force as a function of the distance, a class such so the force is always attractive, but has a minimum not exactly at the bound position, but slightly offset, a profile that has the minimum of, or when I speak minimum, I mean the most attractive, the most favorable electrostatic force at the bound position, soft landing case, which was explained on the previous several slides, namely the electrostatic favors the binding till a particular point, and then at the binding position provides repulsive force to reduce the speed of association. And trivial case when electrostatic is always a repulsive force and the binding is due to some other contributions. And the last thing I like to discuss with you today is implementation of Delphi force molecular dynamic simulation to study a binding of a small ligand to a particular receptor for which we do not know what is the trajectory, and even we do not know what is the opening of the active site. This is a very long story. It comes from our interest of particular disease called Snyder-Robinson syndrome, where mutation in this protein called spermine synthase are associated with intellectual disorders. Another reason that we pick up this protein as a study for our study, as a subject for our study, is that the ligands are highly charged, and the electrostatic distribution, electrostatic field distribution has very prominent features illustrated over here as a kind of electrostatic guiding channels or tunnels towards the active sites. Very quick summary of experimentally known facts. This protein functions only as a homodimer. If the protein does not dimerize, it does not function. The activity is 0. That tells us that homodimerization is very important. This is illustrated in this experimental work, which again, I don't have time to discuss with you in details. We'll be glad if there are further questions. The details of Delphi force molecular dynamic simulation can be discussed here, but again, I'm not going to do that. Essentially, with several words, we take the ligand positioned away from the protein interface, in that case, up to 60 angstrom away from the protein. We use Delphi to calculate what are the forces of each atom of the ligand. And then these forces are given to numbed steered dynamic simulation. And then numbed is run for a particular amount of time. A new position of the ligand is obtained, forces are recalculated, etc. So it's a stepwise procedure till we get some satisfactory results, till we get the ligand inside of the protein. There is a lot to be said about details, and namely not to double count electrostatics in molecular dynamic simulation. And for that purpose, we developed 3 different versions of this protocol. We'll be glad to discuss these things as well. The main reason for developing this protocol was that we were unable with a standard molecular dynamic GB protocol to make the ligand get inside of the protein, no matter how long the simulations were. And this is illustrated in this movie. We said that perhaps this is due to the standard cutoff. So then we increased the cutoffs of calculating both radii and electrostatic interactions to some crazy number, 120 angstroms. The simulations were really very, very slow, but still we saw no success. The ligand never gets inside of the binding pocket. And finally, we applied a Delphi force molecular dynamic simulation, and quite quickly, the ligand gets inside of the binding pocket, which we know from the crystallographic structure. The binding requires rearrangement of quite many loops. And here's how exactly the ligand penetrates inside of the core of the protein. And the thing that I like to mention, when the ligand gets inside of the protein, it is completely buried. A little of caveat. This is perhaps a slide that will be of interest of people working in GB modeling. I'm not sure how much details I should provide in this particular talk, but according to us, there is something not totally correct in some GB models. Consider scenario A shown here, where we like to collect the electrostatic force at particular position of an ion when we charge one ion position at the surface of our protein. And then we simply take the protein and position it now between the source and the position where the force will be calculated. When we did that, these 2 scenarios in GB model of NAMD, we saw a total correlation of results. Namely, the forces are identical despite of presence of this large dielectric cavity. In case of Delphi, the forces, as expected, were totally different. That prompted us to implement Delphi into a steered molecular dynamic simulation. What is the evolution of the ligand position as a simulation time, as measured between as measured as a RMSD of the ligand and crystallographic one? Here we see that in 1 nanosecond, we were able to bring the ligand with Delphi force steered molecular dynamic simulation from 60 angstrom away to a position which is less than 10 angstrom from crystallographic one. And such a simulation were done positioning the ligand in all possible direction offset from the binding position, not just away from the possible entrance of the binding pocket. And again, there is no, from the crystallographic structure, you cannot identify what is the entrance. You need conformation changes to occur. So offset in all position, and these are statistical graphs showing what are the outcomes of different simulations. I'm gonna discuss this thing a little bit, still having about 5 or maybe 10 minutes, depending how tolerant is water. So here we show a case that indicate the RMSD obtained in a long molecular dynamic simulation for apple and hollow structures. And with the green, we indicate the most different part of the structure between apple and hollow protein. And indeed, they are always oriented, positioned near to this putative entrance of the binding site. These are important loops that play a significant role in the ligand recognition. The thing that we probe the same way as explained to you for microtubule binding domain, we see that in apple state, the distance between these 2 loops shown here with the green takes two distinctive states. One distance is about whatever 16.5, another is about 10.5, about 11. However, upon binding of the ligand, there is a prominent peak, and occupancy shifts from predominantly this opening to close conformations between the loops. Again, interplay between two conformations as anticipated to be seen in the induced binding fit. And finally, we attempted to use Delphi force molecular dynamic protocols on protein-protein interactions. And of course, our primary choice was Barnase-Barstar, known to be electrostatically driven formation of a complex. This is the crystallographic structure. And the thing that we've done is to offset Barstar, 30 angstrom away from crystallographic position, and to rotate either Barnase or Barstar along the corresponding axis. And here I'm going to show one particular trajectory when Barstar is moved away 30 angstrom away and rotated on 90 degrees. And here is the evolution of the RMSD between the current position of Barstar and the crystallographic one. As you see from the very beginning, as the time of simulation progresses, we have a drop in the RMSD, indicating that Delphi force molecular dynamic stimulation managed to compensate for the wrong orientation and the distance between the partners. And finally, we end up with RMSD less than 10 angstroms. The question was, and the very same thing is illustrated for different cases. You can read by yourself what are the initial positions. In some cases, Barstar was rotated along X and Y axis. In other case, both Barnase and Barstar were rotated along corresponding axis. And the evolution of LRMSD, IRMSD, and the distance between the partners as a function of time is illustrated here with different curves. If you're not into Capri competition experiment, LRMSD is the RMSD of the ligand, provided that the receptor is perfectly superimposed. IRMSD is the same thing, but just for interfacial residues. And delta D is the difference between predicted distance of the atomic center of the partners and crystallographic one. First of all, we said that perhaps we never saw any of this quantity to get totally to zero. Distance kind of getting very close to zero, but IRMSD and LRMSD, they're always about 10, no matter how long the simulations are. But then we simply decided to run a molecular dynamic simulation of the complex structure, and we got practically the same numbers indeed. Due to the molecular dynamic simulations, the structure deviates from the crystallographic conformations, resulting to non-zero RMSD. So at the end, we were quite happy with the obtained results. And the last slide that I like to show today is again a statistical analysis of numerous run in terms of the distribution of the distance between the obtained and the crystallographic position. The same thing for LRMSD, the same thing for IRMSD. And different curves shows different time windows in the molecular dynamic simulation, relatively the beginning, in the middle, and the final window between 4 and 5 nanoseconds. And I really like to stress on the fact that the simulation length is really very, very short, maximum 5 nanoseconds. And still, we see a tendency, the result, to be clustered at small distances. It is not so prominent for LRMSD and IRMSD, but longer simulation time resulting to a better performance. And I like to acknowledge people that collaborated with us or contributed. The project on microtubule domain was run with a colleague of mine, Josh Alper. The Delphi Force protocol was developed by a former postdoctoral researcher in the lab, now professor at the University of Texas at El Paso. The implementation of Delphi Force into NAMT, namely Delphi Force molecular dynamic simulation, was done by Yunho Peng, who is now postdoctoral researcher at NIH. The development of a protocol studying the evolution of forces in separation of protein complexes was done by Argo, who is now a postdoc with Charles Brooks, and several other students that contributed. Thank you for your attention and acknowledging the funding agencies.
Thank you. So,
do I stop sharing, or you prefer me to keep sharing?
No, in this way it's fine. Yeah, it's good. So while I wait for question to come, I have one myself. When you use the electrostatic part of the coming from solving Poisson-Boltzmann, basically, in a kind of molecular dynamic simulation, there is the remaining part of space of the solvent. How is treated? Implicit or explicit?
What do you mean remaining part?
So you have the two partners, and you are using the electrostatic force from Delphi to calculate to steer the evolution. And the solvent. How do you treat the solvents? Are there explicit water molecules surrounding, or is implicit?
Implicit. Everything was done with a GB model.
Okay, implicit. Thank you. So I see question from Antonio.
Hold on. Now I have to figure out how to.
Okay. Hi, Emil. Great.
Hi, Antonio.
Well, one of my questions was already asked by Walter, but I have a different one. As far as I understood, when you do your simulations, the charge states of the titratable residues are kept fixed. Is it?
Yes, Antonio.
Because I was thinking maybe that soft landing stuff, could it be related to the lack of some kind of reorganization or something like that in terms of charges?
Yes, so we ask ourselves the same question, and we recalculated the PKs of the titratable groups at bound state, and they're all predicted to be ionized. Obviously, when the partners are far apart of each other, we do not anticipate significant perturbation of the PKs, but even in bound state, according to our calculation, they were not perturbed.
Okay.
But it is, this is just one particular example. You're right.
Sure.
In some other cases, the association can cause proton uptake, proton release, and that should be taken into account.
Mm-hmm. Yeah, because we saw that in some cases. It's a different method, of course. It's constant PHMD, but the physics should be more or less the same, right? Yeah. Okay.
We were very interested also in proton uptake, proton release upon protein-protein interactions, and statistically, we see that there is a significant fraction of protein complexes that are supposed to be doing proton uptake or protein release upon the binding, but that comes with a caveat. We assume that the binding in the living cell occurs, let's say, at pH 7, but it's not necessary. Some of these interaction might occur in the lysosome or some other compartments where the pH is different from 7.
Sure. Yeah. Okay, thank you.
I see a question from Miguel.
Hi, Emil. So
hey, Miguel.
It was really a pleasure.
Really
cool. What is puzzling me, I think it was the same that was puzzling Antonio. The soft landing thing is really the first thing that I look at it, we always try to find out where is the cause. I'm sure you did this in the retrospective. So I just wanted to throw at you just one or two ideas that maybe you said, like, yeah, we checked that. So if you think about it, maybe the distance that you correlate to the X-ray is not really the distance that is happening in the simulation. So it means that it's very often that when we stick a protein complex, or even just the protein alone, into the solution and we run MD, we see actually that the protein sometimes swells and then sometimes contracts. And sometimes to take the value per se from the X-ray might be just, we are trying to push something that doesn't like to be. The other idea, and because they are related, the other would be polarization. Maybe there are some polarization factors that will take into account, it will allow for you to actually have that extra angstrom or extra angstroms into the binding that you cannot account with your model. Maybe I would like to hear your thoughts on that.
I agree with you that, and I'm going to use this cartoonish presentation here, and that the trajectory is not a straight line, and we don't know exactly what is the dominant pathway the ligand takes in order to bind to the receptor. And also, again, polarization effect, as you said, might be also important. But the thing that I like and still believe that this might be a real effect is the similarity between, let's say, this curve. Do you see my cursor?
Yeah.
The similarity between this thing and van der Waals interactions. Indeed, at long distance, van der Waals is attractive. At short distance is repulsive. And the goal is really to bring the things together kind of smooth manner.
But maybe our models make the atoms too fat. You think that was my question.
I don't know how to comment on that. I'm not an MD guy, but you know, there are so many factors that might be contributing to that. But I kind of like this biology argument that the soft thing is they should not approach each other with a large velocity, otherwise they'll splash.
Yeah, makes sense. Thanks.
Okay.
Welcome.
I see a question from Marilyn.
Okay, so that was really interesting. So I was thinking, I've been teaching about the tubulin story a little bit, and I'm wondering whether, if you look at mutations that change the degree to which they process without losing a step, so they kind of go 100 steps and then they get lost and then they go another 100 steps, and so whether your model sounds like it might be a good, it doesn't hold the explanation for why you continue to hold on and why mutations or changes change that gripping time, gripping number.
Thank you, Marilyn. Actually, there are two components of that. The first one, we have a paper on how the binding free energy between the microtubule binding domain and microtubule is related to the running distance and the time of being able to stay on the domain. And there is very nice correlation saying that if the binding energy is very strong, that will result of something like a person walking in a mud, being stuck in the mud, but not be able to go so far away. And the correlation is really very nice. And in respect to forces, we have another paper that shows that using electrostatic forces, we can predict pathogenicity of the disease mutations in microtubule binding domain. And indeed, we see that residues that are involved in strong interactions, if they are mutated for some reason, they are found to be pathogenic.
I guess so. That's great. That's I'll have to look at those the next time I teach that. So I don't know if Tom Lowy has his hand up. If he does, Walter can shake his head. But if we have explicit ions, how does that affect what you're? He's really been kind of telling us about ion binding at specific locations, and we don't we put those in as PB ions, they're not very local. I mean, they're somewhat local, but I guess, what do you think?
This is a tough one. We've been dealing with that question as well. Most of the things that I discussed in terms of profile of electrostatic force were taken without presence of ions to avoid this complexity. But we're also very much interested of predicting surface-bound ions, non-specifically surface-bound ions. And it seems to me that electrostatic works very well in this case, namely when the chemistry is not involved. Then the ion binds most of the cases at the most favorable electrostatic potential. However, I don't like to sound stupid, because detecting of non-specifically bound ions is very difficult experimentally, and we judge our success based on X-ray structures. And ions in X-ray structure are subject to so many other things. They might be artifact of crystallographic contacts, the fact that you need to grow a crystal. So I don't want to speak too much about success of electrostatic in predicting surface ions. But if you close your eyes and say crystallographically found ions represent some kind of the universe of the surface non-specifically bound ions, then we are quite successful in doing so.
And one thing that one could do is use the Poisson equation instead of the Poisson-Boltzmann equation and see whether that affected your results very much. And that would give you like a handle on the effects of the implicit ions, which are probably, I think Antonio kind of, or someone has said that they're not terrible.
We actually decided, one of the things that I like very much in our recent work is that we're using so-called Gaussian approach to describe the dielectric properties of everything, the protein, the water, and the interface. And in that case, you ask yourself, how I'm going to treat free ions? There is no border. And then we came with a modification of Poisson-Boltzmann, so we have a dissolution penalty for the free ions. And that results into so-called diffuse interface between the protein, the water, and at least in terms of benchmarking and so dependence of protein binding, it works very, very well.
Thank you.
Welcome.
Okay, are there any more questions? Antonio.
Can I just make an additional comment? It's related with the treatment of the ions. I don't know if it's relevant for Marilyn's question. Maybe it is. One thing we saw in when we compared ion distributions in constant pH MD and with Poisson-Boltzmann, although on the average they look more or less similar, one of the differences is when you go closer to the protein. For some reason, it seems that in the MD simulations, you don't get as much high density close to the protein as you get in PB. For some reason, maybe it's related with the fact that there is some fluctuation of the sides of the surface or side chains of the protein, or maybe it's some other reason. So one of the discrepancies we saw between the PB and the MD was that one. So the ions don't seem to go that as close to the protein. So the distribution at close distance seems to be somewhat different. I don't know if this is relevant.
But Antonio, also that depends on what is your Stern radius in a PB protocol.
My what? Sorry, Emil?
The Stern radius or the Stern layer.
Of course, sure. Yeah, there's, as you said before, there are a lot of parameters, right? So it's always a problem. Yeah, absolutely.
Okay, I don't see any more questions on the Zoom chat. Let me see on the YouTube. Okay, no, there are no questions on the YouTube chat either. So let me thank again the speaker of today, Emil Alexov, and thank everybody for attending the webinar. And we'll see next week with the next speaker, who will be Lauren Webb. Thanks again, everybody.